like to start with this picture. That's picture thing reminds me of me thinking about neurons in the middle of Canadian wilderness. Um, and I might have forgotten to put gas in the skidoo because I was thinking about neurons. There I am thinking about neurons again and receiving an invitation from David and Ran to present at your seminar. I feel very thankful. So thank you for the invitation and the opportunity to talk about these things with you and to hear your questions. Okay, to begin on the story here, I mean, all we are trying to do, in my opinion, is to relate neural data, observation about neurons and synapses with their function, right? Things like attention, memory, learning, emotions. And this is acutely exciting and challenging, particularly these days. I find with these novel data sets that are pouring in, where we're able to record from almost the whole brain and see neurons chatting about. Here's an example of raster plots of different area recorded with those neuropixel probes, and the top one is going through the visual cortex and thalamic areas, and the bottom one is going through motor. In principle, we see everything. We see the animal observing, sensing the world, processing this, and making a decision to make some type of motion and moving, coordinating that motion. So it's tremendously exciting that we're able to see this. I mean, this, as of like 10 years ago, we didn't have that picture. But it's also tremendously challenging. I mean, we look at this and we have to make sense now of this noise, right? And we look at this and we can go on forever at looking at a meaning of potentially the synchronous activity across different areas. Here looks like there is some type of oscillation going on. It's tremendously rich. The main approach to do this is to just treat every spike as being equal and look at the density. So that's kind of a picture of this, where you see the density of spikes across the different areas. That's a manifold that is in itself complex enough. But I've begun to wonder whether this was correct and whether this was forgetting part of what is maybe essential to understand the neural code. And I began to ponder about the role of these bursts of action potential coming at a high frequency that we wouldn't see here at this resolution. There would be a set of action potentials so close together that they almost merge, but they merge into one point. And I think there's tremendous importance in those bursts, and that's what I want to talk about today. But first, what is a burst? So there's all sorts of definitions, and typically you'll focus on the fact that there's suddenly a high frequency. So the intra-burst frequency is high. That's relatively high. There's typically a certain number of spikes that come with this high frequency, sudden increase in the frequency. And there can be a sustained depolarization. The spikes tend to decrease in amplitude, broaden them in width. But then, what is a burst? I mean, typically you just pick up a threshold. You'd say, okay, as soon as I have 2 spikes of 50 hertz frequency, if it's more than 2 spikes at a higher frequency than 50 hertz, I'd call that a burst. And otherwise, I'd call that a singlet. The place where you put the threshold, it can be fairly arbitrary. I mean, we haven't found one way to say this is a burst, and there might not be one. But my take on this is that bursts are actually a way to communicate not the net amount of information that comes in, the net amount of synaptic bombardment that comes into a neuron, but to, on top of communicating this with spike, also multiplex information about the amount of calcium that's in the cell, because calcium is an extremely important second messenger for cells. And if you make a model of the amount of calcium that will be during such a burst, and you vary the number of spikes and the number and the frequency, you normally see that, you know, the higher the frequency, the more calcium there is. And the higher the number of spike, the more there's accumulation of calcium. So that's heat map here is showing higher amount of calcium in the top right corner. And so I see burst as being a way to communicate high amounts of calcium. So you see, you can pick up a threshold, a separation between low amount of calcium and high amount of calcium. That will be a curved line like this in this number of spikes frequency range. And the top one is high amount of calcium, the lower one is lower. And that's what I think, you know, that's what I think a burst is. Now, I think, you know, that there remains arbitrariness in what you say this, what threshold you think this is. But I'm going to leave it at that, because this can depend on the system, and there's all sorts of technical details I don't want to get into. So having, in my opinion, succeeded in evading the question of what is a burst, I'll continue and talk about the burst coding hypothesis. What do I mean by burst coding hypothesis? So I think there's not one, but multiple burst coding hypotheses, which is the fact that a burst means something else than just an accumulation of the same number of spikes, but at a lower frequency or not coming in a burst. You can look at this from the generation point of view, the mechanisms involved in generating bursts, the transmission point of view, and the plasticity point of view. For generation, burst coding hypothesis is something like you need a special or a very specific set of inputs to give rise to those bursts. Here is an example where you need input from two pathways together in order to give a burst. And if you have input only from one of the pathways, you either have silences or single spikes. Another variant of the burst coding hypothesis is transmission, whereby in the absence of short-term plasticity, you would treat every spike, you transmit every spike equally. Every spike is transmitted the same way. You will have a bit of an accumulation when spikes come at high frequency, but they're all treated the same way. This is in contrast with in the presence of short-term facilitation, where effectively single spikes are not really transmitted. You need multiple spikes at high frequency in order to communicate a burst. And in the presence of fast short-term depression, it's the opposite. You do not communicate bursts, and you only communicate the single spikes. So again, here, bursts are something special from the synapse for the synapse. So if this happened, this satisfies this burst coding hypothesis for transmission, and we know these things happen. And lastly, this plasticity, whereby associative stimulation of pre and post side of the synapse will depend on the bursting on the postsynaptic side. And typically you'd get something, at least in cortex and hippocampus, I'll get into this, you'd get LTP when you pair with a burst, and LTD when you pair with a single spike. So these are three different variants. Like, this is encoding burst by special mechanism, this is transmitting burst by special mechanism, and this is being sensitive to burst to engage synaptic plasticity. And I guess there's a overarching burst coding hypothesis is that all of these are aligned, where the burst that is that is generated is also the one that's doing a special transmission. It's also the one triggering plasticity. And this sort of alignment between the mechanism for generation, transmission, and plasticity is seen in the hippocampus. There's very beautiful experiments in the hippocampus showing special mechanisms for generation, transmission, and the plasticity of bursts. For generation, if you look at CA1 pyramidal neurons, these bursts are called plateau potentials. It can be pretty big and pretty long. Will be generated if you have a coincidence of input from the CA3 onto the apical obliques, well, the oblique dendrites here lower, closer to the cell body, and in combination with input from the entorhinal cortex in the apical dendrites of these cells. If you don't have both of those together, you do not see those plateau potential / burst. There's clearly a strong dependence on bursting for the transmission. The hippocampus is a bit special in the sense that most of the connections, at least onto and out of the CA1 neurons, show pretty strong short-term facilitation. And for plasticity, there's really beautiful evidence showing that if you pair presynaptic activity with burst, you get LTP, and that has been shown to not depend precisely on the time, on the coincidence. You can delay the burst by about a second and still get LTP. And that's been shown also to occur in vivo. So it's a very important a series of evidence supporting this importance of burst for regulating synaptic plasticity. And when you pair presynaptic activity with single spikes, you tend to get LTD. And these are all like, here there's really an alignment. There's some experimental studies, for instance, by Takahashi and McGee, that have shown that the burst, when you trigger a burst by generating the two pathways together, you get this sort of plasticity taking place. In the cortex, which is going to be the focus of my talk, things are very similar, although we don't have exactly the same level of precision in the evidence. There also seem to be two pathways. So if you have input that are in the basal dendrites that typically is thought to communicate mostly feed forward information, you need to have a conjunction of that feed forward information with input to the apical dendrites, so it's thought to be communicating feedback information, so maybe about attention or learning or expectations. When the two are there together, you generate bursts of action potentials. There's a very specific circuitry of short-term facilitation, short-term depression, where there seems to be these two subsystems. The ones that are in basal, impinging from the thalamus to the basal dendrites, tends to be short-term depressing. And onto PV positive cells, this tends to be a short-term depression. And then when you connect to this other subsystem, centered on the apical dendrites and the somatostatin positive cells, there's more short-term facilitation. But there's some unknowns here. We don't quite know whether the short-term depression or short-term facilitation onto the apical dendrites. And the plasticity looks the same. You get a sensitivity to burst to generate long-term plasticity, long-term potentiation, and single spikes tend to generate long-term depression. So that's the data. So that's what's special about it. Shows that burst would be special from a bottom-up point of view, and we have a lot of data supporting this. And now, what does that do, right? That's the point where those two worlds can be really far apart. The world of functions and the world of neural data are very far and can be seem really hard to relate the two. But as a field, we've developed this sort of multiple step approach where we can make mathematical models of function. We have mathematical models that's, the world of engineering, the world of AI is making mathematical models of learning, of attention, of different types of attention. And these are well described with mathematics, and these make a more precise algorithmic implementation that we can try to relate with neural data. And I will be trying to relate this not just with neural data, but with computational models of neural data so that we can really make the link closer together. So one possible algorithm that will be requiring another message, something that is like a burst that's able to communicate another message and that will be linked with plasticity, is the problem of the coordination of learning. How can you have synapses that are essential to generate a higher order concept? How can these synapses in the lower order area be tuned to get the right higher order concept? So that's a problem of graded assignment, the coordination of learning. And that's typically in machine learning solved by the algorithm called backprop. And if you take this example where you're classifying images from CFAR, and here's a plot of the learning as you're learning the synapses here according to two algorithms, a global error signal that's saying yes or good or bad. This is pretty bad, doesn't learn very well. And the backprop algorithm that is able to generate a concept of error and backpropagate this error through the network. That is able to solve or to reduce the error in classifying those images. So that's the solution that machine learning has. So what I'm proposing here is that there's an equivalent algorithm that could be built that is modeled along the idea of backprop that uses bursts to represent the error. And these can be using the feedback connections to then travel down the network and represent error in every single neuron. And that will, if you couple that with synaptic plasticity rule, allows you to do a learning algorithm that exploits a specific representation of errors and therefore does something like backpropagation of errors. And the essence of this idea is the idea of communicating two types of information. And how would that work? So that's where we've done some spiking simulations, where I love to show that image every time I'm giving a talk to illustrate how you can use bursts to communicate two types of information, and that the different types of information can pass one another. So here I have a simulation of two-compartment neural model in green and another two-compartment neural model in pink. It's an ensemble of cells. I send an input to the soma. And you see here, the input is time dependent. Now I'll compute the event rate. So I'll discard all the bursts, and I'll only count the single spikes and the first spike in a burst. If I compute the event rate, this event rate is closely matching the input I've given here. Not surprising. This input goes up the hierarchy with short-term depression in the different synapses. So if I look at the event rate of this pink population, I again represent this input. Nothing very surprising. That's like a SynFire chain. But what is surprising is I didn't tell you that in those very same simulations, I was actually injecting a current in the dendrites. There was a current up here that is shown here. And yeah, it's not impacting the event rate, because what it does is it affects the calcium spikes that can be generated through the apical dendrites and tweaks the burst probability. Doesn't affect the event rate at all. So you don't see any effect of the current in the event rate. There was also connections going down with short-term facilitation, and there would be no effect here either. you can see it's not as though the dendrites were too far to be affecting the soma, because if I compute the burst fraction of this pink ensemble, I can recover the input I was injecting to this ensemble. So this information is there in that pink ensemble. And I can actually convey it back to the green ensemble with those synapses. Now, when I'm communicating here with short-term facilitation, I'm communicating only the burst rate, which would be a multiplication of those two curves. So it would look like this. And the burst fraction in red of the green ensemble is following exactly this. So you can represent the information going down in different stages. And now what happens is there's this successive multiplication that happens with the feed forward information. Feedback information is kind of multiplying the feed forward information in multiple stages like this. And I mean, I wouldn't be able to go in all the details, but this is precisely what you need for an implementation of the backpropagation of error, because as error comes through the network, it needs to be scaled by how much this network is active, because that's how much the network can have been contributing to the final error. And that's coupling these mechanisms with burst-dependent plasticity allows you to do the sort of solving a sort of task, the image classification task that I was showing a few slides before. And that's an illustration of, here we have basically 4 steps from linking coordination of learning in an algorithm using computational simulations that are based on neural data. This sort of 4 steps. 3 levels of analysis if we were to use the Mars, not 3, but 4 levels of analysis if you were to use Mars terminology. Okay, so this is stuff that we've published in 2021, and since then I've been focusing a lot on other questions. One thing I want to mention related to this is when we started this work in 2020, and we saw the importance of bursting for communicating 2 messages, we were wondering, what are those 2 messages? The first idea we had was a learning signal, but we also thought about predictive coding. There are papers that were done around that time that clearly showed an alignment between the algorithm that you would normally get for a predictive coding, for predictive coding to work in a hierarchy, and backpropagation of error algorithm. The 2 algorithms can be very close indeed. They can actually exactly map one another under some assumptions. And therefore, you know, we could very well have started and tried to use bursting to do a hierarchical predictive coding model. We didn't do this because we thought it would be easier to illustrate the role of bursting for the coordination of learning, but we think that you could do this. And similarly, this is very connected. Top-down attention. Top-down attention is a type of predictive coding where you're clamping the top predictions. And so top-down attention is, again, very similar, algorithmically speaking, and would require bursting to represent both the prediction errors and the forward evidence. So these are the questions we have now at this point. We're like, okay, first of all, is there multiplexing happening in vivo? And we see something like this. Everybody's only reporting firing rate changing in time. Do we see an independent signal changing as in a time-dependent fashion that would maybe correlate with different things? Maybe it would correlate with errors so that it could be used to coordinate learning, or would correlate with something like attention. And then a second question, or like a third in these steps, is how broad is this code? Can we actually expect all neurons to do this or not? A lot of what we've shown in our 2021 paper was based on the physiology of layer 5B pyramidal cells, and not only this sub-region and sub-cell type, but only the larger version of this and the non-human ones. So if this code is to be important for the brain, it should be more ubiquitous than this. we should have more dendrite-dependent bursting across different neurons. So to address the first question of seeing whether we can see evidence of multiplexing in the brain, I work in collaboration. I tried to look for data that would be supportive of this, and I found this beautiful data acquired by Guy Doron here when he was first working with Michael Brecht and then with Matthew Larkham. And this data is really precious because he went and did juxtacellular recordings next to deep layer pyramidal cells that we know are emitting bursts. And there's no problem with calcium imaging because calcium imaging doesn't reveal a single spike. You lose them, so you can't see when it's bursting. It's typically only the burst that you see, so you can't see the changes in bursting. And a NeuroPixel probe have the potential issue that they might, because of spike sorting, sort out some of the burst, and you don't see the burst anymore. So I wanted to make sure we didn't have those technical issues. I wanted to have those juxtacellular recording. And he had a really interesting task where the animal had to learn to report a microstimulation, so an electrical stimulation given in its sensory cortex with licking, so it gets a reward if it licks within 1 second. So this is an example for a naive cell where it gets this electrical stimulation at time 0. And then you have here this time we don't see the activity, and then if the animal licks within 1 second, it gets a reward, and after it wouldn't get a reward. And then with each of those cells, I'm able to compute the trial average firing rate that will show in black, and the burst fraction, and the trial average burst fraction that I show in red. So this is a naive animal that haven't really seen the task ever. You don't see any stimulus artifact. You don't see the representation of the stimulus. Everything is going to be learned as the animal is learning to go through the task. And so here are a few examples of cells that we found. I picked up a few examples. You often see those, well, maybe 10% of the cells are looking like this. Upon the stimulation, the firing rate jumps up to really high values and then goes back down. And the burst fraction follows the same thing. The two signals happen to be correlated. So if all the cells were like this, I would conclude that there's no multiplexing in the brain because the two signals are highly correlated and just mean they just communicate the same thing. But there are other cells like this one where the two are the cell, there's first inhibition in firing rate and then a jump in the firing rate and with a delay, and the burst fraction just jumps throughout. And just decay slowly. So the two go up together, but they don't follow exactly the same time course. Other cells would look like this, where the firing rate goes down upon stimulation, and the burst fraction jumps up and remains high, higher than the baseline for a while. So here the two signals are completely different. And here's another example where the burst fraction is almost constant and the firing rate just goes down. But these are just exemplars, but it shows there's a bit of everything under the sun, and it's not as though this is noise. I mean, the burst fraction is sustained up. It does stay up. If you take all the cells that are responding with an increase in burst fraction and you average them together, you get this picture here, where there's a jump of 20% a jump of maybe 15% in burst fraction, while on average, these cells basically have no change in firing rate. So there's this huge signal that is there. It hasn't really been, it's time dependent, that hasn't been studied very much. So I conclude from this that there is possibility for multiplexing different types of information that could be occurring in vivo. Now I wanted to look at specific correlates of errors and attention. So first, error. So the animals were sometimes missing they didn't lick at the right time. The stimulus is pretty strong, and they're pretty expert in this at this stage. So we think they've sensed the stimulus every time, but they missed the lick within 1 second. And if you compare the firing rate within for hits for the full line and miss for the dash line, to look for a representation of error, whether the response of the cells depends on having or missing or hitting the reward, you don't see a significant difference. That's typical. I mean, people have tried to find a modulation of hit and miss in the representation of the inputs in primary cortices, and they typically don't see any difference. He's not at a level of firing rate. But then they typically look at the beginning of the response, because the firing rate after that dies off, right? But what happens if you look later? After all, like, the animal really knows it made a mistake about this time here, where it really is too late for it to lick to get a reward. And you see here that actually in the firing rate, there's an increase in the firing rate in the misses. So that when the animal misses, there's actually an error in coding that comes in half a second after the end of the response window. And then we can look at the event rate, so kicking out the burst, or the burst fraction. You see a fairly small representation in the event rate and a pretty big representation in the burst fraction. That comes in sharply right at this time here, about half a second after the response window. So that's there seemed to be a representation of errors. Now I wanted to look at something like attention. Now that's not, the perfect task for this, but what I did is I separated the responses in terms of both firing rate and burst fraction of all the cells here. They are aligned with the high firing rate on top, keeping the same ordering for the burst fraction. Then I've separated all those cells according to the number of days the animal has been under the rig, and has been learning gradually to complete this task. So I've separated the first days and days 3 and 4 and days 5. And what you see is there's a temporal evolution of the burst fraction. The burst fraction is coming in late for the first days, earlier for the later days, and much earlier, but although for a smaller portion, for the last day. And that is what makes the firing rate go higher and higher at this time, because as you have more bursts early on, in the small portion of cells that increase their firing rate, you have more firing at this time. And I see this as, because the animal is gradually becoming better through these days, I see this shift in the burst fraction as a correlate of how the animal gets better. To make this explicit, I quantify the timing, the bursting moment. So doing kind of a center of mass, but for these curves, for day 1 and 2, day 3 and 4, day 5, and so on, I calculate this moment of the burst fraction according to day training times, and I see a negative correlation. So the burst fraction moves to an earlier time as the animal gets better. So there's a strong, significant correlation or negative correlation between the time of the burst fraction signal and the training. And if I look at the event rate, there's no correlation. So the event rate, so the net number of single spikes and initial spike in the burst, do not come earlier. But the animal does get better in those times. I could correlate that with behavioral accuracy, and it was also showing a significant correlation. And the firing rate does show a net correlation. So although this is preliminary, I think it shows there's a potential to have a real study that is aimed and built to look at this. There seem to be two representations, one that comes late that represent the error, and one that would be consistent with top-down attention, although a lot more has to be done in that direction. And we're trying to see how we can make sense of these observations and see whether there's a way to implement a learning rule and relate one with the two signals together. This is work in progress. So I've talked about those 2 points now. There is a representation of error in attention, or something like attention, that is multiplexed. Now I wanted to talk about the question of the properties of neurons. And so for this, I'm changing. We took a different approach, a more fundamental computational neuroscience, classical computational neuroscience question is, what is the input-output function of a neuron? And can we think more about the role of dendrites in order to see whether there's an effect on bursting that's very general? So typically, we characterize input-output function of a neuron. This, by the way, is work by a PhD student in my lab, Zach Friedenberger, very talented PhD student. And he is like, okay, we typically look at leaky integrate and fire neurons for an input-output function or direct injection of current in the cell body of a cell. And you'd get something that looks like this, where you characterize a firing rate as a function of input strength and the amount of noise. And what you typically see is that you get a nonlinear relationship. And as you increase the input strength, there's more firing rate. And if you increase the noise, well, you linearize that curve. It becomes more and more linear, although it's still, rectified linear. Okay, so that's the first order. That's when you calculate the average firing rate. But there's also the second order, which will compute the CV, so the coefficient of variation of the intervals, where CV will be high, where it's very variable intervals. So in other words, the neuron is bursty when CV is high. And we want to understand how the CV depends on the input strength and the amount of noise. And this has been studied a lot in the past, and typically, as you increase the input strength, the CV goes down because you get into this mean-driven regime, and the interspike interval becomes more regular. And this is, again, linearized by noise. So as you include noise, you have less of that going down happening. Okay, it seems like we've understood the input-output function, but that's for inputs to the cell body. What happens so that's only when you're injecting input here. What happens if you think about inputs going through the whole dendrite, right? And in this, there's the world of dendritic computation has been focusing a lot these days on the idea of an additive modulation of the input-output function, where input to a dendritic compartment can be processed non-linearly, a bit like in an artificial neural net. You have a non-linear subunit that then feeds into the soma and comes in additively, so it shifts the FI curve. This is the famous view of neurons as a multi-compartment neuron that Jutta Poirasi has developed. This is certainly happening, and actually we've worked on this, and we've just a parenthesis here, it won't be long. We've worked on making machine learning benchmark with different number of compartments within our hidden units, and trying to see whether adding dendrites makes it more powerful, better for learning. And we see that indeed adding dendrites, at least in some cases, improves our accuracy at the task we're doing. But closing the parenthesis. The other type of changes that has been developed in the literature is that of gain modulation, where your net input to the dendrites actually changes the gain to the neuron. So that has been done experimentally by Matthew Larkham. But what happens for the second order? What is the input-output function in the presence of dendrites, or for the CV as a function of input strength? We don't really have a theory for this. So we wanted to understand this because the CV, as I said, is related to bursting. And it's fairly easy to do this. We'd either take a multi-compartment model and then simulate a synaptic bombardment, and then calculate the CV as a function of the net input strength. To compute the CV, we need to do multiple repetitions of like long averaging, and so we were looking for a quick way of doing this. We thought about effective compartments, where you think of a model, a two-compartment neuron model, like I've shown in the first prop picture. But we were thinking, it'd be nice if we could use the older model of the Green's function of the dendrites, right? That have an analytical formula for predicting how much voltage you have as a function of input distributed in space. That was developed by Winfried Rahl and sometimes with John Rinzel. Because because this is, you have one formula that predicts the voltage, and with the voltage, you could use escape rate and have these renewal theory methods to compute the CV, or the interspec interval distribution, in one integral. It would be so much quicker. We didn't have to wait for averaging over long instances of multiple instances of noise. But the problem in the Green's function, we can't really use it as such because it's for a model with passive dendrites. And we know that the dendrites are not passive. They produce all sorts of dendritic spikes. So Zach then decided to extend this and include the excitability of dendrites into this Green's function model of the dendrites. So in the past, we would be, if we were to use a Green's function, we would have the voltage predicted as a linear filter, which is our Green's function, of the input, spatial temporal input, time dependent and space dependent. That gives us a spatial temporal voltage. We might add adaptation to this, but adaptation that's only in the soma. That was the previous picture that we could make. Now he extended this with this picture where we have the Green's function for integrating the inputs, but we allow for a somatic spike to also move in space and back propagate to the rest of the dendrite. And we also allow for dendritic spikes to be emitted and forward propagate to the cell body. So that was an exciting for us a modeling approach. I like that because for me, this is a one equation model of a dendrite, of a neuron with dendrite, and ability for neurons to have active dendrites. And not only this, is we could use this model, and here I'm injecting current at one location and current at another location. This is what the Green's function would tell us if the current don't change in time as a function of space. And then we can calculate the probability of interspike interval in just one line, a very complex line, but a one line. And this is the theory here are the colored curves, and the black curve is what you'd get if you average over multiple instances of noise. There are here multiple cases, the presence or absence of backpropagating action potential, but it's not the point I want to show you. So we could use this theory to then calculate the input-output function. This is the firing rate as a function of input strength. In the absence of dendrites or passive dendrites is the dashed line here, and with active dendrites we have the blue line here with different strength, with the paler line being stronger strength. And the dots here are Monte Carlo averaging. Well, our theory is just following the Monte Carlo very well, so we could focus on the theory in the future. And what you see here, you see this sort of bit of gain modulation due to dendritic input, and certainly a shift, so the additive shift that was the two models of dendritic integration that I mentioned earlier. Now I can look at the second order, and I can look at the CV as a function of input strength. Now we saw something that I didn't expect at all. Typically the CV, like in the dashed line here, is a passive case. CV typically drops with input strength. More input, more regular intervals, more into the mean-driven regime. But that's not what we saw. We saw an increase, and the increase in the CV that is controlled by the dendritic input. So the higher dendritic input, higher CV. So I was like, oh, what's happening? At first thought this was a bug. But then we looked at this, and I think for the sake of time, I'll go a bit fast on this. We looked at these changes, the first order changes, characterized by the shift A and the gain G, and we look at this effect on the CVC across different conditions for the size, so the duration of our dendritic spike and the amplitude of our dendritic spike. And we see that at the additive effect on the first order picture is there whenever you have big dendritic spikes. And this effect on CV is there very often too, whenever you have big dendritic spikes, and pretty much for any duration. And the effect on the gain is not so frequent. Now we wanted to understand this. Okay, it's very consistent. It happens no matter what sort of dendritic spike you might have. And by the way, you know, the calcium spikes are more in this region, and the NMDA spike more in this region. This will be the sodium spike in the dendrites. We typically understand this in terms of the transition from the fluctuation-driven regime to the mean-driven regime. If in the absence of noise, we have this nonlinear relationship with no firing below threshold, below Rio base, and firing after, that's nonlinear. In the presence of noise, there can be firing the fluctuation-driven regime because the fluctuation are driving the firing and not the mean. And when you transition to the mean-driven regime, then the interspike interval is very regular. The CV follows typically this. You drop in CV as you transition to the mean-driven regime because the interspike interval are driven by the mean just pushing you to the threshold regularly. So we understand this increase of the CV as a function of the input strength that is tuned by the level of dendritic input as a transition between mean-driven regime and fluctuation-driven regime of the dendritic spikes themselves. So we say, you know, if the input's very low, then when you have dendritic spikes here illustrated in the right, they will come close to the threshold, but not consistently reach the threshold. So the dendritic spikes themselves remain in the fluctuation-driven regime. But of course, as you approach the fluctuation-driven, the spike threshold, you start having to generate some spikes here that are separated by the interdendritic spike interval. So you have an increase in CV that comes to kick in at this point. And then in the burst regime, where more consistently, when you have a dendritic spike, this dendritic spike is above the threshold. And so then in this regime, you typically have just maybe a few spikes in between the dendritic spikes, but then burst coming in every time you have a dendritic spike. But then the next stage is that of full mean-driven regime, where both the mean and the dendritic spike levels are above the threshold, which is a case of very regular firing. And so as you approach that other transition, it drops down. And so that's how we understand this CV increasing and then decreasing. And so we think that with this sort of process, different types of dendritic activity, not just those calcium spikes that are in layer 5B pyramidal neurons, can be used to do something like multiplexing. But of course, more work needs to be done to see how pervasive this code can be. And that's all I wanted to say. I've showed you that there's evidence for bursts being different than single spikes, that these can implement algorithms like backpropagation of error algorithm, prediction of error, predictive coding algorithms. And there is some level of evidence supporting this in vivo, and that more research onto the mechanism generating those bursts, particularly in understanding better the input-output function with active dendrites, would probably help us seeing how pervasive this code is. So with this, thank you, and I'm looking forward for the questions.